Thank you for coming. It's my pleasure to introduce Nick O'Neill. He's a graduate student in our lab. He started graduate school here in the fall of 2016. The only way I can remember that is by looking in my phone and seeing when I got the first email from him. He's taken an interesting path to where he is today. He has training in logistics, and he worked on model systems before starting graduate school, so he has a diverse background. He's very passionate about spinal cord injury and using the very best outcome measures to evaluate whether the various treatments we are studying in our lab actually are useful or meaningful. He's going to tell you about a project that he's been working on for about last year or so. And he's been thinking about it ever since he came back from the Ohio State Spinal Cord Injury course. What he's doing, I think, is so cool. I keep videos in my phone and I show them to people at the airport when I'm waiting for airplanes.
Thanks, Vance. Hopefully everyone will be as enthusiastic about this as you are after they see what I'm going to talk about. So my name is Nick O'Neill. I'm a member of the Limbics Lab, and the Limbics Lab is made up of John Bixby and Vance Levin, and also Fusion with Hassan Al-Ali. And so the work I'm going to show you today wouldn't be possible without the help from everyone in the lab, but I want to single out two people in particular for their contributions. Carmen Ma, a postdoc in our lab, helped immensely with building the machines that you get to see later in the talk, and also Victor Jan, an undergraduate computer science student who spent time in our lab and whose work was crucial to getting the machine learning aspects of our work up and running. And also thank you to my committee for helping guide my work in a rewarding direction. I want to start by talking about one of the ways that Dr. Lemon and Dr. Bixby convinced me to join their lab. Something you likely know about them is they always seem to be just ahead of the curve with regards to technologies that get adopted by everyone else in the field. Just a couple of years later, when they were describing what the lab does to me, they told me about this concept of high content screening. High content screening takes advantage of improvement in robotics and automated analysis in order to get a really unprecedented amount of data from our in vitro cell culture assays. So how exactly does this happen? You start with this image of a single neuron, and there's quite a bit of information that can be gathered from an image like this. You can see the amount of neurites that have grown out of the cell body. You can see branching happening. You can see a neurite extending away from the cell body. And if you have antibodies, you can also see whether the absence or presence of a protein you might be interested in is there. The next step is to capture all of this data from multiple neurons. So we've now zoomed out to see all of the neurons that you would see in one field of view. This is the step where automation really increases the amount of data that we can get from these neurons. An algorithm is run over these images, and the features that we care about, such as neurite outgrowth, are automatically measured and quantified from all the cells in that field of view. A benefit to this automation, in addition to increasing the amount of data that we capture, is that we've been able to remove some of the experimenter bias from the analysis. This same algorithm is run over every cell regardless of the condition. We then zoom out again to the well view, and you can start to see how more neurons are becoming part of the data set. And so if we zoom out again, now we have all of the wells that would be included on one plate. And finally, we see how this method allows you to replicate this process across multiple plates and multiple weeks into a full experiment. And this demonstrates how this method is scalable. We've increased the throughput, and by doing so, we've also improved the statistical power of our experiments. The next step after identifying growth-promoting treatments in cell culture is to test these treatments in vivo. And this is where the true benefit of adopting a new technology like high content screening really comes in, because we're able to test so many compounds with this method that we can prioritize compounds to test in vivo based off those we think will most likely have a significant effect. And indeed, using this method, the lab has identified treatments that cause axon growth in vivo after spinal cord injury. On the top, you see the spinal cord of a control condition animal, and on the bottom, extensive growth of axons past an injury site in an animal treated with a compound based off of results that we saw when it was screened. So after seeing how successful this method has been, one of the things I started to think about was whether we could apply the similar concepts of capturing a lot of information, reducing experimenter bias, and increasing throughput to other areas of our research. So one of the many nice things about working at the Miami project is you get to see all the interesting work patients are doing for rehab and physical therapy when you walk in downstairs. And so if you're like me, you tend to spend a lot of time thinking about what it means really to recover lost function. And the best way that we can design our behavioral assays so that our experiments in the lab reflect that. So a question I had was, are we meeting these same standards that we outlined for high content screening with our behavioral recovery assays? So this is a video of a mouse performing an assay known as the pellet reaching task. Pellet reaching is a well-established way to study fine motor control and recovery of forelimb function after a cervical spinal cord injury. When the lab started to try and implement this assay for our experiments, I thought back to those standards from high content screening. So you can think about this too. Does it seem like this method captures a lot of information? Possibly. It certainly captures some, but you can see from the video that the primary outcome measure that you would be limited to see was whether or not the mouse grabbed the pellet, but any more advanced information about how exactly it moved to do so would be difficult to assess. Is experimenter bias minimized in this assay? Certainly not. That is my hand, you can see, placing the pellet in front of the mouse, and so I'm basically completely integrated into the experiment, and it'll be very difficult to separate any effect I'm having on the performance of the animal from its performance on its own. And third, is this scalable? I've heard from Vance that there is almost nothing an undergraduate research volunteer won't do for a letter of recommendation to medical school, but I think there really is a finite amount of times that you can place a pellet in front of a mouse like this with forceps. So naturally, this led to a second question. Can we develop an automated and quantitative method to assess subtle changes in reaching behavior after a spinal cord injury? The problem our lab faced is that our behavioral recovery assays fail to meet the same standards as our in vitro assays. And so our proposed solution was to take a similar approach the lab had used for high content screening and automate both the data acquisition and data analysis phases of our behavioral recovery assay. So for the rest of this talk, I'm going to cover the steps that we took to try and solve this problem. The first step was to automate the presentation of the pellet. In order to do this, we got in contact with the lab of Kareem Fowd. His lab specializes in rodent behavior and rehabilitation strategies after injury. And we were able to use designs his lab has made to present pellets to rats and modify them so that we could build something similar for mice in our lab. This helps address the problem of experimenter bias. The machine presents pellets on its own, controlled by a repeating timer, and once the mouse is in the box, no further interaction from the experimenter is needed. Vance and John were kind enough to indulge me after I made the case about how beneficial these machines would be for our experiments and purchased a 3D printer for the lab. We could then print multiple of these machines to use in our in-house. This addressed another problem by making the assay more scalable. We could now have multiple animals performing the task simultaneously. The benefits of this automation became apparent when we saw how many more trials we were able to get from each mouse using the machines. When done by hand, we would get around 20 trials that were usable on video for each mouse in a 10-minute testing period. And after we linked all the machines together through these circuit boards that control the position of the arm and a repeating switch that allows the arm to go back and forth in a set amount of time, we're able to get around 75 usable attempts per 10-minute testing period. As you can see here, time lapse of this video. So while I can watch over the animals happening in this phase, I don't have to physically be there interacting with them at all while they're performing the task. The next step was to automate the analysis of the videos. And again, we found we were just in time to adopt some cutting-edge technology that would allow us to do this. In order to automate analysis of the videos, we decided we wanted to track features in the videos. And we were able to increase the resolution of our videos to the point where individual digits could be identified, like you can see in this image. The points we decided we should track were the nose, the pellet, what I'm calling the thumb, which is just a nub on a mouse, the edge of the dorsal surface, farthest away from the thumb on the ulnar side of the wrist, the index, middle, pinky, or ring and pinky fingers. So at a conference last year, another lab showed us this new machine learning software called Deep Lab Cut. Deep Lab Cut is a convolutional neural network developed by the Mathis lab, and what it allows you to do is track anatomical features from images without the need for any specific marker being physically placed on the animal. So what we do is we take this image, we mark what we would like to label as shown on the previous slide, and we give this data to a neural network. And what we get back as output is the predicted X and Y coordinates from those features in new videos we haven't manually marked yet. The first thing we wanted to do was if we could use this X and Y data to determine whether in video reaching attempts, where in the video reaching attempts actually occur. And so to show you how this is done, I'm going to show one frame of video at a time. On the left, we have our video with the markers overlaid on top of the video. And in the middle, we have those markers plotted by their X and Y coordinates within the frame. And on the right, I have a column color-coded for each marker. If that feature is present, in the frame, a dot will appear in that column. And each row is a single frame from the video. In this first frame, you can see that only the nose and pellet are present. Therefore, a measurable attempt hasn't started yet. We now move to the next frame. You can see in the video that the mouse's paw has just started to appear, but since we're using the X and Y data from features as our output, we don't start identifying an attempt just yet. Now we see a paw clearly appear, and an index finger has been identified by the software. You can see the index finger appear on the chart, and index is marked as present in this row on the right. The presence of any feature other than a nose or pellet marks the start of an attempt. You can now see that the thumb has also appeared. More fingers and the back of the paw start to become visible as the mouse wraps its fingers around the pellet. This frame here is what we identify as the apex of the reach attempt. You can now see the mouse start to pull back its hand with the pellet in between its fingers. And as the mouse paw moves back, you start to see different features leave the frame until only the nose is present again. This marks the end of an attempt. So, now that we have this matrix of data representing an attempt, I wanted to know whether you could use the patterns that you see there and see if they were distinguishable from the patterns of a failure and the patterns of a success. So this is the trajectory plot of all the coordinates from a successful attempt displayed over the time of the attempt. So we wanted to see, again, for another set of a matrix of XY data, if we could do the same thing for a failed attempt. What became apparent was a big distinguishing feature between successes and failures was clearly the tracking of the pellet, as you can see here. So based on the data you just saw in those plots, we used a machine learning algorithm known as a random forest classifier to sort data sets into successes and failures. After it was trained, we would provide a matrix of values like you saw earlier, shown on the top. And the random forest would use a variety of randomly generated decision trees to predict whether the data set came from a successful attempt or a failure. In addition, we had to classify reaches that were made when the mouse made an attempt, but there wasn't a pellet present. So you can see we used the random forest classifier to predict whether the data set represents a success or a failure. We found that the random forest classifier was highly accurate at classifying attempts into reaches, successes, and failures. If you look at the first row, actual reaches, they were predicted correctly 95% of the time. In the next row, true failures were also correctly predicted 98% of the time. In the bottom row, true successes were predicted correctly 85% of the time. What we found was that for some successes, the paw had completely grasped the pellet, and therefore the pellet was obscured from view. And sometimes when this happened, the algorithm believed that the pellet was dropped rather than retrieved back with the paw. I doubt you could ever get this to 100% accuracy because of this problem in the videos, but this number, 85%, has increased as we increased the amount of sample data that we gave to the algorithm, specifically when we gave particularly difficult frames into the training data set. So I believe this number should continue to improve as we add more data. We were happy with this data, but this still hasn't gotten to one of the main reasons why we decided to develop this assay. We wanted to know more specifics about not just if the animal was able to perform the task, but how. And there's an implied question there that we can make specific. Does a binary scale, such as success, failure, capture what we mean by recovery of lost function? In order to address this, we need to look at function at a more fine-grained scale. From previous studies, we know that reach and grasp in rodents can be broken up into 4 broad categories of movement. There's the aim and advance. The animal pronates its wrists when it grasps the pellet. The fingers wrap around and grasp the pellet. The paw will then supinate, and the arm is withdrawn for retrieval of the pellet. Work from several labs over the past few years has identified which level of corticospinal tract projections to the spinal cord are important for each phase of this behavior. Neurons that terminate in the C2 to C4 region are involved in every phase of this reaching movement. In contrast, neurons terminating in the C6 to C7 region primarily contribute to the rotational wrist movements, pronation and supination, as well as digit flexion. Because of these separate contributions, we hypothesized that if we injure the spinal cord at C5, we should be able to detect specific deficits in pronation and supination while the animal still retained some of its overall ability to retrieve the pellet. In the rodent, fine motor skills are controlled by the corticospinal tract, but other tracts, such as the rubospinal and reticulospinal, contribute in a more prominent way than in humans or non-human primates. For this reason, the injury we performed primarily cuts the corticospinal tract, seen in blue, but also damages, to a lesser extent, the rubospinal and reticulospinal tracts. I want to point out that in the past, if your lab wanted to identify a specific deficit in skilled reaching behavior, you were likely limited to a qualitative assessment. For example, does this animal display abnormal pronation or supination? Yes or no. These are some example images from a rat that has a specific reaching deficit. The lab that performed this experiment are experts in rodent behavior and have members of the lab who are also physical therapists, so they can assess deficits from images like this. But if we want to make this assay something that any lab that studies spinal cord injury can do, and that's my goal, what we need to do is have a way to measure the changes in reaching behavior that is quantifiable and doesn't rely so much on expertise. To measure pronation, we decided to focus on the two features on opposite sides of the dorsal surface of the paw. And this first image comes from a moment just before the paw turns over and pronates to grasp the pellet. You can see that the thumb is visible, but because the hand is in a neutral position, you can't see the other dorsal edge. Because the paw has pronated, we can now see the opposite edge come into view. And as the paw pronates, the distance between these 2 points in the x-plane will increase. To visualize this, I put a black bar in between the 2 points that shows the distance just in the x-plane. At the moment of pellet grasp, the paw is as pronated as it's ever going to get during any point in the attempt, and you can see that at this point, the distance between these two features has also increased. As the pellet is grasped and starts to be brought back into the box, you can see that the wrist starts to supinate as well, and so this distance in the x-plane starts to become lower again, trending back in the other direction. And you can see, once again, when in order to draw the pellet all the way back, what happens is the animal usually returns its paw to a neutral position so it can fit its hand and its pellet back through the slit in the box where it's reaching out of. And again, the dorsal edge now disappeared again. So there's no measurable pronation in this image. So this is a slow motion video of an animal that's been trained on this task for a while, and it hasn't undergone a spinal cord injury. And this is a successful reach and grasp. And what I want you to focus on looking for is those 2 points, the thumb and the dorsal edge of the wrist, and see if you can notice how the hand turns over as it goes to make this attempt. Excuse me. So what I thought I could do was plot this value that was represented by the black bar, the x distance in between these 2 points, to measure the amount of pronation over the course of the full attempt. And so this green line is representing the value of that x bar over each frame that's shown during this attempt. And as you can see, the whole length of this attempt is less than 200 milliseconds. So the only reason we were able to capture so much of this information was by using a camera that was recording at 240 frames per second. So like I told you, the injury that I wanted to do, I believe, was going to cause a deficit in pronation and supination. So I performed that injury on these animals, a cervical C5 hemisection, and what we noticed was they exhibit less pronation during successful grasp. So again, if you focus your attention on those 2 points on the back of the wrist, this animal is still succeeding at the task, but you can see in this video that it looks like there may be some noticeable difference between how it's performing the task before and after the injury. And so when I was first looking at this, I thought that seemed like something I could tell by looking at the videos, but the reason I wanted to make it quantifiable was because it would be impossible over lots of videos to decide just exactly how much of this function had been lost. So now I'm going to plot the amount of pronation measured the exact same way pre-injury for this animal post-injury. And this is the same exact animal before and after an injury.
In thinking about baseline,
Oh, these animal success rate is about 45% of the time. And in order to get there, it takes somewhere between 10 and 14 days of training.
14 days. Now, are they mice that train well and versus mice that do not train well?
Yes. So the only mice that you'll ever see in a video that I'm showing are ones that reached about a 35% level of success rate before an injury.
35%.
And that number sounds pretty low, but across many labs do the single pellet reaching task. It's actually fairly common now if you're looking at cervical injuries, and so that's right in line with what most people see for mice. For rats, it can be a little bit higher, a little bit smarter, have better skills, but for the mouse, somewhere between the average of 40% in a distribution across all your animals is pretty common.
And do you do left versus right limbs?
Yes, so when we're training each animal, I take notice of which hand it prefers to grasp with, and the pellet gets moved off center to that side to encourage it to use one hand or the other. So like in this example, the pellet is moved to the right and the hand is reaching with the right. So as the animal gets better and we move the pellet more to the right, it wouldn't be possible, given where the slit is in the box, for the animal to use its left hand to grab this pellet. Just the angle's too difficult. So the only animals that get included in the injury model are ones that have a specific hand preference, and then that's the side hand that we injure. So afterwards, right after the injury, within the first week, the animals don't really display any ability to successfully grab the pellet. They may attempt sometimes with the opposite hand, but they kind of figure out that you can't, because of where you place the pellet, use the other hand.
Nick, what does the delay mean? There's a slight delay in the injury pattern.
So the way that this is set up, keep in mind that as you move this way, this doesn't mean supination past neutral, even if it's going the other way. The baseline all this way down is neutral. So how I plotted these was start.
Yeah, delay.
I'm good. I'm good. So how I plotted these is I take the apex point of the reach and go backwards to the first point where a feature shows up that can be used to measure this. So in order to see this line, you need both of these points in the video. So let's say this is across the frame, it takes 10 frames to get through here. It's more than that, but for example. And 5 frames in is the point where you grab. If both of these are grabbing at the same time, there's only 3 frames before where the animal's pronated enough that I can see both features. So both the attempts start at the same time, but I can't start showing anything until it gets around this level where both the dorsal edge and the thumb start to show up.
So they're pronating later.
Yes.
Right?
So
That
it's-
actually means something, right?
So these attempts are, to the best of my ability, to match the apex of the reaches across these, all starting at the same time. So the absence here, it's still attempting, but you can't measure anything yet because it's like just at this point here where you see fingers coming out, but it hasn't started to pronate yet.
Nick, for the software that you're using to track the points, do you have to, how accurate is that software?
It gets better the more work that you put into it and the better quality videos that you have.
And do you have to train it for each animal, or it identifies the digits?
So the biggest benefit to us was increasing the resolution of these videos and recording at a high enough frame rate that there wasn't that many blurry stills in between each phase of the attempt. And so we labeled a couple hundred of these frames this way. And so what we did was across maybe 10 animals or something, take 25 frames from each animal, each one of it during an attempt, label the features, and combine them into one data set that the algorithm trains on.
It's two different things, right? It's the software that's identifying the points, and then you have your separate classifier then that takes that information and does more with it. And so I'm just wondering, like, that first step, like you have to do stuff for your classifier for identifying the features and
So the
classifier, but that before all that happens, you've just got this software that you're using, and so.
to get these markers, these markers came from training on more than one animal in our set. So I did a combination of left and right, probably about 6 animals of each, like I said, and 25 to 50 frames from each animal. So you get, and in there, because I needed to track specific things like you were saying in the next step, included in the training data set were examples from successes and failures. So there were frames mixed from times when it reached without the pellet, times where the pellet dropped away, times where it was brought back. All those things I tried my best to include in the training data set. But now, if you can take this algorithm as trained and just put an animal from right now that was not part of the study in it, and it will track the features to this same capability. And they did a lot of really nice fancy work with the algorithm that's kind of cutting edge machine learning stuff, where they changed the resolution to higher, lower, they move the frame left and right, all sorts of randomization. Once you include your training images, it does a bunch of randomization on that to improve the robustness of the algorithm. So if I happen to have a video that was a little bit less clear, it still succeeds at getting that because on purpose, the algorithm has blurred some of the images and tried its best to still see if it can track during those.
So the inputs to your classifier are just like on the XY position of each.
It has X, Y, and the confidence of the algorithm in that specific XY point. So like that matrix I showed, I take basically that square that has X, Y for each one, confidence for every one of those cells, and those are examples.
If that represents the time at which the computer can detect the 2 dots, then shouldn't all of them start at that second column? So in the first column you have the 2 dots in a vertical axis, right? And then the second column is time point, is a position at which now the 2 dots start to appear separate. So that's the case, and shouldn't the pre-injury and the post-injury all start at that second column?
They should be fairly similar. There should be, even though the widest gaps in between pre and post injury are here in the center, like we're saying, there's parts where this comes that should overlap back with the post injury. And so we do see that, like you're saying. It's just that in reality, the difference between up here and here is not that different in the video.
Have you thought about adding a second camera from the side?
So we're in the process of doing that. We can use the same exact process that we did for this, using 2 cameras to get this plotted in 3 dimensions. And so we should be able to capture things a little.
It's getting expensive for cameras all of a sudden.
Have you looked at like how the other digits are acting differently? So if you take the other dots, right, and look at the distances between them, because at least I can see from the videos that this injured one is picking up a little bit differently than the prior to injury.
So that's spot on. We were thinking the same thing, that we would see some differences in maybe the spread of the fingers and also in how the paw supinates on the way back. I'm going to show these side by side so we can talk about it. So we did do those analysis for these 2, and for both the things that you mentioned, finger spread, and then if you watch closely in the post-injury animal, on the way back, it supinates more than, it's kind of subtle, than the pre-injury animal does in order to not have the pellet fall out of its hand. And so I didn't include the data for those because they're less clear than this, but there is a significant difference. It just isn't as clear given the data from this animal.
And it's so clinically relevant.
And how much variability is there within a single injury level with respect to the strategies that are used?
There were some animals, this could be simply, I'm doing these surgeries manually, so there's could be just a consequence of exactly how severe I am performing the surgery. But there's some animals where if I went a little too deep or too far laterally and hit more of the reticulospinal tract, they're slower to get this ability back. So like this animal is from 2 weeks after the injury, and there were some that were at this point still unable to perform successes. So they exhibited similar behavior of not turning the wrist over, but all the videos I have are from failed attempts where it's completely missing the pellet. So just to show that there was a difference in pronation in successes, these are the videos I included, but the same behavior applies in failures, and there's some animals that just fail to successfully grab pellets after the injury. So I plotted a good set of pre and post-injury successes from the same animal, exactly the same way, an even number of pre-injury and post-injury successes. And what we saw was that on average there's a distinct difference between the amount of pronation that the animal was able to perform pre-injury and post-injury. So we have a quantifiable way to separate how much pronation has happened as a result of this injury.
Is all of your analysis down at the hand? Have you done any like eye gaze, okay, or head positioning to see if there isn't
A third camera.
Well, no, cameras are cheap these days.
So that there is lots of,
Would you mind? I'll come talk to you about that. Okay.
you are with this software really only limited by your imagination. So, and camera buying, like Matt said. So depending on how you want to position this camera, there's a lot of things also about the shoulder, elbow, head track, like sniffing the pelvic floor and stuff that you could track. In order to get the videos that look this good, we have to place the camera 2 centimeters in front of the pellet. So you're kind of limited logistically by the setup on exactly what you can video. So if you try and come from the side, we can't get a really good angle of the shoulder, say. But if we could position the camera in a way to do that, there's nothing stopping us from using the same exact method we did for this to pick your behavior, whatever you think is interesting, apply the same methods, and you can track the features.
Because the Denver Fote did something like this in humans when she was here, and she was also looking at the way bodies changed, body positioned, kind of preparation for the limb moving, as well as what was happening actually at the site of the grab.
When you watch by eye, there's stereotypical behavior that happens with the mouse before this hand actually shows up in the video, right? Positioning its body to go for an attempt. And so I haven't quantified a difference between those in this injury, only again based off the limitations of videoing one thing at a time. But it's certainly something that we could look at.
There's an entire eye gaze strategy that's within sports medicine that looks at judges for athletic competition and how they make a plan for moving their eyes to capture the most information. Here, obviously, you've got a still target, but it might be interesting to move the target. Move the target.
Whether the animal's gaze would adjust based off if we adjusted where the pellet was.
You're giving these graph noise too much credit.
It's possible.
85% of the time.
Until we do some specific way of separating this in the study, I'm really unsure of exactly how much sight versus smell versus muscle memory contributes to their ability to grab the pellet in the same location each time. So each one of those things could be looked at separately.
Is the pellet real food?
Yes, it's a 20 milligram kind of cereal pellet.
I'm sorry, you need to continue, but do you put them on a low diet or anything like that?
In order to be encouraged to keep doing this, they're put on a food-restricted diet. So the very first day, we take away the food and we replace it only with these kind of pellets until the animals say, okay, this new pellet that's not the standard thing we give them here in the building is also food. But then once that happens, we continue to give them the regular food just at a moderated level and check the weights to make sure they're not losing weight over the course of the experiment.
And one more thing, I mean, over time, rehabilitation can be very good in terms of upper extremity function. So they're kind of going through a rehabilitation strategy themselves doing that. If you looked at, do you plan on looking over time after injury and see if they improve?
So that's exactly the thing that I want to do is because now we have this method of performing hundreds of attempts versus in a way that we couldn't do that before for a bunch of animals at the same time, and we can analyze the videos this way, it's possible for us to look at a lot more time points and also to just give them more training too in a way that wouldn't be possible if we were doing it by hand. So I do want to do that. That's my goal.
No electromagnetic stimulation.
Forget about biologics, go through, okay, good.
Quick question. Is there a way, since you mentioned some of these very fine, movements and differences that you can measure, you will test probably generating therapies, I'm assuming. But you have variability that you can simply do
if I'm remembering correctly, Jay did this in a past life for lesion size compared to deficits you see in certain behaviors. And so I think that we probably should do that afterwards when we're doing histology on the animals. The lab that gave us this idea for the machines did something very similar, and that's why we got the idea to use that injury in specifics. They took the spinal cord and looking at it like the face on a clock, made a 1 o'clock injury, 3, 5, 6, and determined which of these gives you a behavior that's not 0 on this and that's nothing. So what they said was attempting to make that quarter size injury in the spinal cord is what gives you a reliable deficit. And so that's what we're trying to match. So we're going to do the same thing that he did. If some animal stands out as having 0 in this, we can look afterwards at the lesion size and say, oh, okay, it's because I went past the quarter size injury in that animal.
You can 3D print a surgeon.
No, Lance has a lot of money, you can buy the Da Vinci robot to do this surgery.
I think you're maybe confused lab resources.
So I just want to say, and it looks like there's less post-injury attempts here, but that's actually because this animal succeeds a bunch of times and there's no frame where both dots show up. So it's able to reach out, keep its hand neutral the entire time, just barely hang on with fingertips and cusp it back into its hand. So this is something that we never see happen pre-injury. So pre-injury, there's no attempts by an animal like this where it doesn't at least try and turn its hand over somewhat, but we see that regularly with the after injury. So as was just suggested, what we want to do in the future is obviously use this assay to test our best axon growth promoting treatments and see if we can shift where those trend lines were back towards the pronation ability of an animal pre-injury. So to summarize, I want to leave you with kind of 3 key ideas that come from the way machine learning and automation have improved our behavioral assays. So by doing the experiments this way, we've increased the amount of information and detail that can be captured. We've increased the scalability, permitting experiments with higher statistical power. And we've minimized, to the best of our ability, experimenter bias in both the administration and analysis phases of the assay. And that's it. Thank you.
This would be a great time to have this, clinical to preclinical discussion about making sure questions we're asking in the clinical arena right now with rehabilitation and upper extremity function and now be asked more elegantly in this very beautiful way to look at corpus spinal tract function in mice.
So they wanted me to repeat the question for the video. So Dalton's question was, we know a lot more about the specifics of deficits in the clinic because of all the patients we have downstairs, and we may not be doing previously behavioral assays that reflect that in a way that's most likely to lead to clinical translation. So that's kind of the goal of doing this, is to see how well can we match what we think we're going to do for human patients after spinal cord injury with what we're really testing in our functional behavior recovery assays in rodent models in the labs.
And what Nick has not mentioned is that in the last few weeks, he's come down and he's watched participants working with our physical therapists and doing different reaching or grasping assessments and gripping and different functional tasks. So he's there, he's thinking of it, and he's really trying to incorporate what we're doing into the way he's thinking.
So Katie's been kind enough to let me observe some of what we do downstairs, again, for the same purpose of seeing what are we doing with humans, how best can we match that to our experience upstairs.
So, do you have any plans to maybe take the machine learning technology and apply it to other behavioral assays like we do?
Lindsay wants to know if we can take this same methodology and apply it to other behavioral recovery assays. The answer is yes. Me personally, I probably am going to focus on this and getting the supination and the other things that were suggested, finger extension, and again, once we have three-dimensional video, looking at the same features that way. But in the lab, we've also tested this out on water maze tests and other things, and so if you have the videoing capability in your lab, anything that you can video, we can try and measure using this method. And so the videos that you saw today all came from commercially available GoPros that you could buy at Best Buy.
Just since this is being discussed a lot, there's a startup, a neuroscience startup that builds these cubes, and it has cameras in all orientations, so it records everything, and then they let the deep learning algorithm extract all features, including how often the mouse takes naps, like everything you can imagine. And then the software is able to cluster drugs based on any behavior, so things that cause the same, even shaking movement, temperature, how often it takes naps, how it moves. They are able to cluster these drugs or mechanisms in a really nuanced way. So like Nick said, anything you can imagine could be analyzed that way.
So Hassan was really responsible for, when we were thinking about the high content screening, making that have multiple parameters that we could all measure at the same time. And so as he's saying, for this behavioral work, we could do a very similar thing, where if the significance of a single thing isn't strong enough, we could have a bunch of behaviors that record all the same way and combine that data to reflect a general distinction between different injury models.
Can you go back to that graphic?
Yeah.
So based on that, so what would be recovery? How would you define recovery based on that graph? Are you gonna target the animals that are just straight up and down, those vertical bar graphs on the left, or is the target going to be the one that show deficits?
So what's interesting is this is all one animal. So every one of these lines comes from one animal. I have information from this from multiple animals, but I just wanted to include all the successes from one. And so the way that these successes are happening both come post-injury. So you can include these in something where you would measure an average or a mean across this, because it's outside of the data that we can capture. But we have two options. We can see whether the average amount of pronation starts to move back towards the pre-injury level as far as max amount of pronation that happens, or also what percentage of total successes, move out of this category. So in this animal, about half the time that it succeeded, it didn't pronate at all. So 50% of the successes, it grabbed in a neutral hand position. So if we have a treatment that says, oh, it continues to succeed, but now every time it succeeds, it looks maybe more like this, and it's escaped this ability to have no pronation, I would see also that as a significant recovery.
All right, so even the vertical bar, those are successes.
Everything is a success. So that the animal has just performed successes, but they look distinctly different than it did pre-injury.
So when you talk about treatments, you're going to do it on a per animal basis. You're going to compare one animal.
So the patterns of this, this is a general pattern we see across animals, but because there's so much here, I think the safest way to do it would be to have its average amount of pronation before and after an injury.
So how do you make the comparison between a treated and untreated mouse broken or a cohort? How do you integrate all this information in one value that you can, how are you going to do that you can compare multiple groups there?
Either of the two ways I suggested across all the animals. So you could say across our 10 animals that we have sham and 10 treatment, how many of those moved back towards this line and to what percentage? And I keep doing this, sorry. Same exact way across all animals individually. First, for how many of the animals are we able to shift these successes out of the no pronation phase?
So what happens if you have equal numbers of successes and failures with and without pronation of this? Like, do you have to pronate to improve your success rate?
I don't know. As you would suspect, losing the ability to pronate usually hurts your success rate, right? So I included the same amount here, but the true success rate in these two that isn't shown, this is like 40 and this is 30 or some 25 to 30%. And so I included the same amount of successes to put them next to each other, but the animal has lost a little bit of its ability in total to succeed compared to how it was pre-injury. So I wouldn't suspect you would see it go in the opposite direction, where if you lose pronation, you then get better, but it's possible, depending on, I don't know, there could be individual differences by animal. They clearly can succeed in a way that looks completely different than they do normally.
You have to measure something that directly correlates with success rate as well.
You can use, so most of the time when you use this assay, the main readout is success rate. And so that we could capture that as well. So that, like Hassan was suggesting, getting multiple parameters, if you include, for each animal you have its success rate change, its pronation change, its supination change, et cetera, and those can all be aggregated together.
If you take only the successful trials, is there a time difference between how long it takes to get the food back to the mouth?
These tend to be longer because you can see in this video, the animal either by ability or maybe sense, because they lose some sense that we're not necessarily measuring in the fingers either. So it can sense when it's grabbing the pellet, and so it holds on to be more confident in grabbing it. I'm, anthropomorphizing a little bit into the mouse, but as it feels grabbing this pellet, it takes its time bringing it back. So these actually generally do take longer, and it's the reason why you see a longer thing, amount more here is because it's holding into this position.
You just want to get the food in the mouth, so you're doing this,
Right.
rather than sort of like taking your time, making sure it's grasped, and then
Mhm.
making your success rate higher.
Mhm.
Like that's what you're aiming for.
But, so, if as they learn to grasp food in their unorthodox way, they can get back up to do the same number of attempts pre-injury, but in a different way. Are you considering those successes? Or, I mean, so they can learn to grasp, but with their, inherent function, they adapt, pick it up this way, right?
That's why we want to, exactly. Yeah, that's why we want to get multiple, the whole reason we get multiple ways to measure this is if we were just looking at success failure, we wouldn't know that. So it's like in the videos I showed at the very beginning, the animals definitely are compensating. They're doing a different pattern and they're still succeeding. So without looking at it this way, that would be hidden to us.
Right, but even if they do, they still do, so as long as they do it in the right way, by even only 2 times, say, that would be a recovery, you would consider that a recovery function?
I'm unsure yet. We haven't done enough yet to decide exactly what's going to qualify as it's possible due to, happenstance in its arm hitting the slit or something as it reaches out that the hand could turn over. I would need to see more than a single time to say this is pronation. But if there was any sort of repeated change, then yes, I think so. Thanks for all your questions.